In Salisbury, Massachusetts, at the mouth of the Merrimack River, there's a salt marsh, perfect for the analysis of certain environmental conditions. Like all salt marshes on the east coast of the United States, this one serves vital ecological functions and connects our coastal freshwater streams to the ocean. It was the aim of graduate students Mark Miranda and Chris George of Antioch University, New England, to analyze the effects and relationships between the salt marsh area, pH, bulk density, and macrofauna. Seen here in brief, we set out to analyze the effect of alkaline seawater and the crushing weight of tidal inundation in this fascinating landscape. This video will show a little about the salt marsh environment, why we chose our research question, and show some of the actual field conditions while collecting data for a report. A salt marsh is a tidal estuary where fresh water from the land flows into the sea. It is a wetland where accumulated organic matter forms headbanks of peat, which is histosol soil. Seen here, all estuaries need a barrier beach in the distance, blocking the direct effect of the ocean water so that the soft, peaty soil can accumulate here. The headwaters of many salt marshes in New England terminate in human inhabitants. Landward, the high marsh community, is dominated by tall grasses when in season, and abuts a forest community. Fresh water from inland headwaters, here restricted by a culvert, is of a lower pH than seawater, which is actually alkaline, averaging around 8 to 8.1 on the pH scale. Looking out towards the sea, you can see the barrier beach in the background. Along this creek, from low to high marsh, tides will increase, seen here in this chart from high to low on a daily cycle. Inundation, low tide. Here are some images of a little bit in between. Mid tide, where inundation is incomplete, and a little bit lower, but still relatively flooded. One great ecosystem service of the salt marsh is a buffer against harsh weather and storms. But the salt marsh also enjoys fair weather. Let's look at some biologic factors of the salt marsh. The soft substrate of the salt marsh is home to varied and numerous infaunal organisms, including mollusks, like this periwinkle seen here, and numerous other shellfish. The grass layer in the high marsh is a microhabitat for macro arthropods that also dwell in the soil, like this decapod. Burrowing crabs also inhabit the surface layer and act as bioturbators. Seen here are two common salt marsh species, and underwater, invasive green crab. Here is an interesting soil-dwelling biotic factor, the hole of a burrowing woodchuck. This nearby dead fisher cat is a good sign of the intermediate nature between land and water. She might have been after fish, but probably didn't mind taking a look at that groundhog either. This is the Salisbury salt marsh at the mouth of the Merrimack River. The pyramid ahead is an aid to navigation to signal the entrance to approaching boats. As can be noted ahead by the darker zonation on the rock jetty beneath the aid to navigation, the waterline may come up about 10 feet on a high tide. That's a lot of weight to put on this soil. Our interest is what is the bulk density of soils that are closer to the mouth of an inlet versus those in the high tide zone closer to the land where they get less overall inundation. We are also concerned with pH along the high to low tide gradient, along with biologic factors that may influence soil density through bioturbation. Let's take a look now at the site. All quadrats were laid at sample sites to analyze the existence of infaunal organisms. In some instances, we had to lay the quadrat on the grass or in the thick mud, like this so-called black mayonnaise mud seen here, noted by its color. In these plates are examples of infaunal organisms that were dwelling in our survey area, including worms, more worms, evidence of crabs, and some macroarthropods, known as amphipods or scuds, and insects. Samples were retained for bulk density and pH analysis to answer our questions about how these factors would change along a creek from the seaward margin towards the landward. Field conditions were excellent, with blue skies as we set out on April 13, 2016, to collect our data.